Okay. So let us see where we are. We are coming up to exam number 2. So exam number 2 is going to cover lymphatic/immune, so both of those chapters or whatever, and it's going to cover our respiratory system. So just those two. That's coming up this weekend, this Saturday, so that you can get that out of the way and then have a relaxing spring break. But let us review some points for what we went through in respiratory system already. What are some important things that you think that we were learning and going over last time? Whether it's topics that we were talking about, things that you had questions on, things for the exam objectives, if you were looking at those, hopefully. What do we think? Let me scroll through to see. Or just what did we talk about? Yeah, Maddie. Thank you.
Can you explain how the pressure gradients?
Yeah, so that's a good one. So we're talking about pressure gradients. So that gets us into the fact that we had 4 processes that we said that are going to be important. You know me, as a teacher and as a parent, I'm always going to shove a little bit of extra nutrients and brain food at you. So 4 processes. The first one was ventilation, which is what we had gotten into yesterday, or not yesterday, but last time, and what we're going to review this time. And that has to do with pressure gradients, but we will also find that there are 3 other processes, right? So there is external respiration. So this is gas exchange between the alveoli and the pulmonary capillaries. So this is actually getting carbon dioxide to come out of our bodies and oxygen to come in. That also works on a pressure gradient, but slightly different. Then we have gas transport. So gas transport is going to be getting it from the lungs out to the cells that need it and back, right? So we got to get it to and from. And then finally, we have internal respiration. So internal respiration is also gas exchange, but this is totally within the body, that's why it's called internal, between the systemic capillaries out at the tissues and those same tissues, right? So this is getting the oxygen out of the blood into the tissues, picking up their carbon dioxide waste products, getting it into the blood so we can transport it back up to the lungs again, okay? So these 3 are what we're going to go over today. When we're talking about pressure gradient in terms of ventilation, what we were saying is that there were, let's write that down, pressure gradient, gradients and generating them. So a gradient means that it's going to be high someplace and low someplace else, right? So there's going to be a difference. And like anything, whether it's diffusion or something like that, or a ball rolling downhill, it's going to go from high to low. So in order to get it to move from one place to another, whatever it is, whether it's air overall, whether it's oxygen, whether it's carbon dioxide or something like that, we need to generate a gradient so that it'll go from high to low. And hopefully low is going to be where we want it to go to, right? So in terms of ventilation, we said that there were 3 pressures involved, right? So the first one was called intra, that's within, intrapleural pressure, right? So intrapleural pressure is going to be that which is in the lung cavity itself. And the main role for that is it's going to be adhering the wall of the lung to the wall of the cavity, so that when our muscles contract, like we talked about last time, like external intercostals and diaphragm, when they move the walls of the cavity, the lung moves with it. Okay? That's its main role. So that one, we always want it to be like a negative pressure. We always want it to be lower than our other pressures so that we kind of maintain that suction, so it stays stuck to that. pleural cavity wall. Okay, so there's intrapleural. Then we have atmospheric pressure, which is not something we control. And then we have intrapulmonary pressure, which is sometimes also called intraalveolar pressure, because that's the pressure inside of the lungs or inside of the alveoli. Now that is one that we can control, right? And that was what we were talking about in terms of ventilation of the lungs. So when we change, we said that there's this relationship between volume and pressure. So when we change the volume, there is an inverse change in pressure. There's kind of an opposite change in pressure. So when we, for instance, when we're doing inhalation and we increase the volume of the cavity, that drops the pressure. That drops the intrapulmonary pressure. Because the volume gets bigger, so the pressure eases off and gets lower. Okay? Once we have intrapulmonary pressure that is lower than atmospheric pressure, now we've generated a gradient. So once it is lower in here compared to out there, even by 1 millimeter of mercury, because remember we're talking pressure, and so that's the units for pressure, millimeters of mercury. Even when it's 1 millimeter of mercury less down here compared to out here, we're going to get a gradient, it's going to move in. Okay? Likewise, we said for exhalation or expiration, either one, right? Same difference if you want to call it that. What we're going to do is we're going to shrink the cavity, right? We're going to let a lot of those inspiratory muscles relax. They're going to come in, the walls are going to come in. Imagine if you were in here and the walls started coming in, right? So just like we talked about the walls of the heart, the chambers of the heart, when those walls come in as the heart contracts, that generates an increase in pressure. And the blood in there is like, I got to get out of here, right? So same thing in terms of the molecules in air that's within the lungs. So when the walls come in, when those muscles relax, the pressure is going to go up. Once the pressure inside of the lungs, intrapulmonary pressure, once the pressure inside of the lungs is higher than atmospheric, now we have a gradient the opposite way. So it's high here in the lungs relative to low out here, and it's going to leave the lungs instead. So we're generating that pressure gradient. We're making the intrapulmonary pressure higher or lower than atmospheric in order to drive movement of air into or out of the lungs. And the way that we do that is we use those muscles to change the volume of the cavity, which then changes the pressure. So the volume, we change the volume, which then changes the pressure and generates that gradient. That's a good question that kind of touches on a whole bunch of things, right? But mainly, it's explaining ventilation. So if you got a question, it's like a short answer question on ventilation, like how does it work? What are the muscles involved? What are the pressures involved? Things like that. You're going to need to name all three of these pressures and discuss what their importance is, right? So that one, sticking the wall of the lung to the wall of the cavity. These ones is where you'd want to be talking about generating a pressure gradient, where we change the intrapulmonary pressure by changing the volume, getting it above or below atmospheric, which draws the air in or out. So we had a bunch of stuff in there. Anything else? Thinking. How about this one? There is an exam question, an exam objective that is a tracing. So in those exam objectives, when I say trace or follow or something like that, in this case, like a molecule of oxygen through all the different parts of the respiratory system, from the time you inhale it until it like goes into the blood, then what we're talking about doing is listing the different anatomical structures. In this case, that molecule of oxygen would pass by in order to reach its destination. So we'll see these coming up more and more, right? So we've got a urinary system we're going to go through. We're going to talk about fluids going through different things, right? We've got a reproductive system that we're going to wrap up with at the end of the semester. Again, where we're going to be talking about the movement of gametes or sex cells through different tubes and things like that. Digestive system, again, following food through a big long tube. And it helps us organize our thoughts in terms of what are the different parts, what's the order that they come in, and then being able to, now we've identified the parts, and then we can start thinking of, okay, why are all these parts there? What do they do along this pathway, etc.? Okay? Okay. So, in terms of a tracing, this one, as I mentioned, will ask you to trace a molecule of oxygen from the time that you inhale it until it goes across into the blood in the pulmonary capillaries, right? So what we want to do is we want to start with where it starts to come into that respiratory system. So we're going to start with, you could start with, for instance, you could say the nares or something like that, or you could say the external nares, the vestibule, etc. Where we're kind of starting. We're starting with the nasal cavity. So then we have our nasal cavity, yeah. After we get past the opening, we go into the space. Then it goes into the pharynx, right? And remember that the pharynx, there are 3 sections. We start out with the nasopharynx. That goes to the oropharynx as we're working our way inferiorly. That goes into the laryngopharynx as we get down towards the larynx. Ta-da! See, we're doing good things. All right, so we get to the larynx. When we get inferior to the larynx, where do we go? Where does the air go after the larynx? What's the air tube? The trachea, yeah. So now, right, part of the role of the larynx is to separate the food and drink from the air. So now we're tracing a molecule of oxygen, so it's going to go into the trachea instead. Later on, when we do the digestive system, we could start, some of these are going to overlap in terms of like the pharynx and stuff like that. But now we're doing the air passageway, so we get to the trachea, and now we get to the bronchi. So remember bronchi, plural for bronchus. We've got primary, secondary, tertiary bronchi, right? So you can kind of just list those all together. So you can go primary / secondary / tertiary bronchi. Okay, so we've gotten through those. Then from the bronchi, what do we go to? Bronchioles, right? Like arteries down to arterioles, we go from bronchi to bronchioles. So there's a couple different types of bronchioles, right? So we have what are called terminal bronchioles. And if you want to do that slash thing like I was doing before, you could do that as well. Terminal to respiratory bronchioles. Those respiratory bronchioles, when we get to this level, now we're actually at a thin enough tube where we can actually start exchanging things. So we've gone from our conducting zone to our respiratory zone. So respiratory bronchioles to alveolar ducts, which is what we saw in the microscope. Remember, very thin walls, so definitely can get across there. To the alveoli. Now that question asks you to trace it until it gets into the blood. So we've gotten down to the alveoli, which is the smallest structure where we're actually going to do exchange in the lungs. But what we have to do is finish up by talking about that what's called the respiratory membrane. So the respiratory membrane is what oxygen and carbon dioxide are actually diffusing across in order to get into and out of the blood. So that includes, and however you want to write it, so you could write respiratory membrane and then write these structures. I'm going to run out of board that you'll be able to see. So that's respiratory membrane. Pretend I'm still writing over there. I'll write it over here. So the respiratory membrane is, and you can simplify it a little bit as well. You could say the alveolar wall, which is simple squamous epithelium, across the fused basement membrane. And then across the capillary wall, right, which is also simple squamous epithelium. Okay, so that's the respiratory membrane, those 3 parts right there. So by that time, after we've gotten across the capillary wall, now that oxygen is in the blood. Okay? So this is a listing of the anatomical parts that you would want to list in a short answer question, right? Just in that order. So make sure that you have all of the different parts, right? So the 3 sections of the pharynx, the different types of bronchi, the different types of bronchioles, etc. Now, we're not having you do all 23 branches of the bronchial tree, but you at least need to include those 3 types of bronchi, 3 types of bronchioles. Alveolar ducts, alveoli, and then the 3 parts of the respiratory membrane. And you can write it out just like that if you want, right? Or you could write the word two, or you could put like a little dash or something like that if you're typing it out, if you get that question. Okay? Does that seem all right? Okay. All right, so I think that's pretty good for a review, unless we have other types of questions. Let us take a look at these other things. So we're finishing up with ventilation. So that's what this and the following figures are going through. We didn't really talk about what's called labored breathing. So when you're breathing really heavy, well, let's back it up. When you are just doing kind of a resting breathing rate, when you're doing that tidal volume in and out, like you are right now, then you're mainly using your diaphragm and your external intercostals in order to ventilate the lungs. So they contract and they are going to move those walls out. And then when you exhale, you really just relax those. So as they relax those, the walls come down, the diaphragm, the floor, if you will, comes up. And that's enough to generate that pressure gradient. So it's only if you start breathing heavy, deeper, etc., would you actually include other muscles as well. So if you really need to, whatever, blow out candles or something like that, then you're going to need to recruit other muscles as well for forced inhalation, forced exhalation, or this heavy breathing. So what happens is, not only for forced expiration or exhalation, not only are you going to relax the diaphragm and those external intercostals, but you can recruit the abdominal muscles. And that's why sometimes when people are doing the spirometry, when they're measuring tidal volume, if you feel those abdominal muscles being contracted at all, you're probably doing too much. And that's maybe why you were getting too high of a value for tidal volume. Okay? You really just relax those other ones. You really shouldn't feel your diaphragm going, or your abdominals going. But if you need to, right, you recruit those abdominals. Think about when you're blowing out a candle. So that's going to force some of those abdominal organs up against the diaphragm. It's going to push on it even more forcefully. And there are internal intercostals now. So internal intercostals are between the ribs. They have an opposite fiber orientation, so they have the opposite action. They basically forcefully pull on the rib cage to pull it down as opposed to just letting it come down on its own. Okay? For forced inspiration, if you're taking a big breath in, if you're doing that inspiratory reserve volume that we measured in lab, you might recruit other muscles like sternocleidomastoid. Remember that one, right? So we're going to try to increase the volume of the cavity up here instead. So sternocleidomastoid, the scalenes, which is not one that we learned previously, but you can see how it attaches up here. Some of our upper ribs, the scalenes. And then pectoralis minor might be involved as well. So those are trying to raise the roof, if you will, right, this way. So we have external intercostals doing that way, diaphragm doing this way, and now we're recruiting these, right? And that's why if you're taking a big breath in, you can't be all slumped over like this. You have to be straight up and then, right? It's not just that you're opening up your larynx and things like that. You're recruiting some of these other muscles, too, up associated with the top of the pleural cavity. All right, so those are going to be involved in forced inspiration when you're taking a big breath in and really trying to get more volume. So what we can see is that with those muscles changing the volume, then we see the pressure change as well, right? So if you were doing, for instance, just our tidal volume, you might see a half a liter, right? A half a liter in, half a liter out, something like that in the order of 4 seconds. And what we can see is during that process, we see the pressure change. So this is pressure relative to atmospheric pressure in millimeters of mercury. And notice that we see this increase and decrease only by about 1 millimeter of mercury in our intrapulmonary pressure. Intrapulmonary pressure, that's within the lung itself. So at this point, when it's negative, we see our inhalation up here, right? Because we're drawing the air in. Here, where the pressure is positive, this is where we're blowing our air out, right? Because it's higher in the lungs compared to outside. So this is relative to atmospheric. So we just create that gradient. And it also works to, again, keep the lung wall attached to the wall of the cavity itself. Okay. The next little section has to do with just reviewing what we talked about in terms of spirometry. So that means in lecture you would also have to know those different lung volumes or capacities, what an average value might be, and where you could find it on the graph itself, right? So if we gave you this graph and there were no labels to it, would you be able to label tidal volume, inspiratory reserve, expiratory reserve, and things like that? Not today, but by Saturday. Yeah, that's okay. Yeah. All right. So tidal volume, remember, is going to be about, right? About 500 milliliters, about a half a liter of air. So we see it right there in the center. Inspiratory reserve volume is going to be that extra amount that you could inhale up above that, about 3 liters. Expiratory reserve volume is that extra amount that you could exhale after your normal tidal volume. That's about 1 liter. And so if we add those together, inspiratory reserve and tidal volume and expiratory reserve, we get this, vital capacity, right? So vital capacity, we see kind of those separations here. Vital capacity is the maximal amount that you could take in or blow out in one breath. That's the maximum amount that you could actually exchange in one breath. That is always going to be smaller than total lung capacity because you're going to have that residual volume that always stays in there, about 1 liter of air that stays within the bronchial tree, keeps those alveoli from fully collapsing and things like that. Okay? Okay. Now there's certainly a range to these as well, so you don't have to have that exact number. If you want to choose a number that is, approximate, that's going to be fine, but there is a range to that as well. So there's a plus and minus, at least 100 milliliters or more of air, depending on your body size and things like that. So I wouldn't worry too much about this FRC, the functional residual capacity, or the inspiratory capacity, but the ones that we focused on before, like we see here. Bless you. See, now that took abdominal muscles. That was inspiratory reserve volume that she took in there. Some expiratory reserve volume. So when she took that big breath in order to try to blow these things out of the respiratory system, right? That's the whole idea, is that you want to take a big breath in, blow it all the way out, right? So that would have been a good example of vital capacity right there. I didn't even pay Alex to do that. She just did. Okay. So this section is just wrapping up a little bit associated to do with those lung volumes. As we were talking about, there's going to be that residual volume that stays in there in terms of things that are within the respiratory passages as well as in the alveoli. So that kind of sums up that residual volume as well. So nothing new in terms of that dead space there. It sounds a little bit ominous to call it dead space. It is interesting, though, that when you take a full, fresh breath of air in, the air that is going to be at the alveoli is not totally fresh, right? So you have this residual stuff that's stuck in there, about 1 liter that gets stuck in there, and then you're getting this full volume of air from the external environment that kind of mixes with that. To a certain extent, right? It's still going to be enough to produce a gradient, enough to exchange oxygen during that breath and things like that. But it's not totally efficient because we got to keep those respiratory passages open. So there's a little bit of that kind of stale air in there as well. Okay, so from this point, what we've done is we have covered ventilation, 1 out of the 4. But now what we're going to do is we're going to talk about external respiration, gas exchange with the external environment. We'll then talk about gas transport, then internal respiration, which to a certain extent seems like a lot, but here's the deal. External respiration and internal respiration basically work the same. It's just the location is different, that's all. Okay? So once we understand how we exchange gases between the alveoli and those capillaries, then we'll just apply it with those systemic capillaries out of the tissue, and you have that one already done. Okay? So it's not too bad. And the other good thing is that gas transport, we already have a step up on that one because that's going to be a role of the blood, and we already have a foundation for understanding how gases are transported in the blood. We're going to review that from what we learned before with the blood, and so we're already building on something we know. So that's nice. Okay? Okay. So there are some physics principles in here as well that we're going to need to understand, but we're not going to need to necessarily memorize in terms of names or anything like that, right? So we had Boyle's law before. Didn't care about the name Boyle, but we do care about the fact that there's this inverse relationship between volume and pressure. That was important, right? So now we're talking about Dalton's law here. So Dalton, one of Patrick Swayze's best roles, outside of Point Break. So that was Roadhouse. I don't know if you've seen that one. Yeah. Anyway, we'll make it his homework, right? Who is Dalton? Dalton had to do with gases in a mixture. So the deal here in terms of what's important in terms of respiratory system is that when we're breathing in air, it's a mixed gas, okay? Now, of that mixed gas, we are interested primarily in oxygen and carbon dioxide in there. But in terms of pressures and concentrations, if we want to think about it that way, when we have a mixed gas, each gas contributes its own pressure to the total. So what that means is when we look at the total pressure, like atmospheric pressure or something like that, like we see here, generally atmospheric pressure we're taking it to be about constant. Now, like we said, it does change. Like when we have rain and gets a low pressure system or a high pressure system and things like that, it does change for sure. But we're kind of just assuming it's about the same, about 760 millimeters of mercury, or sometimes it's called 1 atmosphere, which makes sense, right? Because there's actually 1 atmosphere sitting on top of you right now, right? The weight of that, which can be ominous, but that's sitting on top of you. So that's the pressure that you're dealing with. That's the concentration of these gases that we're dealing with as well. So in that 760 millimeters of mercury, there is one portion or part of that from oxygen. There is a part of that coming from carbon dioxide. There's a part of that coming from nitrogen gas, and a part of it coming from vaporized water. Now, there could also be other things in there, right? Hopefully, there's not going to be a carbon monoxide in there, but there can be other gases in that mixed gas, right? So each one of these contributes to the whole. Interestingly enough, most of the air that we breathe is actually nitrogen, like 79%, something like that, most. But we don't use gaseous nitrogen. In fact, there are only certain organisms that can take gaseous nitrogen and turn it into an actual solid molecule to get it into kind of the food chain and things like that. They do nitrogen fixation. It's kind of like a general biology thing. But in any case, each of these contributes to the whole. So for instance, if we wanted to calculate the partial pressure, right, the pressure just from oxygen, its part that it plays as part of the whole, you would take the total, 760 millimeters of mercury, multiply that by the percent of oxygen. So normally we would consider oxygen to be about 21%. So of all of the gas that we are taking in, 21% of it is oxygen. So 21% times 760 gives us 160 millimeters of mercury of oxygen in this air that we're breathing in. So that's the partial pressure. That's the correct term for it, is partial pressure. The pressure that gas contributes to the total. You can also think of it, I kind of think of it as concentration, right? That's the concentration of oxygen in that particular volume, in that inhaled air that we're talking about, okay? Okay, now Henry's law, this one, again, don't care about the name. This one is also talking about the fact that, this gas that we're inhaling, it's going to be going from a gaseous form, it's going to be going into a liquid. So we have to be able to get it into that liquid, and that is going to be dependent on not only the partial pressure or its concentration, but also its solubility. So how well it goes into a fluid comes into play, right? Whether it's easy or hard, that's going to come into play if we're considering temperature as a constant. So that one, yes, it's very true, and it's something that you should know, but we're not going to go into a lot of detail about that. So the solubility of that particular gas also comes into play in terms of getting it into the blood. But let's take a look at this overall in terms of these partial pressures. Partial pressure of oxygen as well as carbon dioxide. So this figure that we have here represents the full respiratory system to a certain extent. At the top, we see the pulmonary circulation up here, okay? So we see there's the alveoli of the lungs, and these are the pulmonary capillaries. Down at the bottom here, we see the systemic circuit instead, right? Between the heart and the tissues of the body. So each of these that we see here represent a little bit of a graph Showing us concentration. I don't know. Somebody backing up outside there again? Okay. It's not somebody's pacemaker going out. We don't have to worry about the AED. Starbucks, what?
It's garbage day.
Oh, garbage day. I was excited about coffee. They were doing delivery or something. Oh, by the way, I'll have to send out an announcement about it. But Wednesday of this week, which is the 8th, Student Life is doing like a big event. I think they're calling it their March Madness event, where it's going to be kind of like carnival themed and stuff like that out in the Saguaro Mall. So there's going to be like the whole like caricatures and like carnival games to play and stuff like that, and free food and snacks and giveaways and things like that. So that's going to be Wednesday. So when we get out of class on Wednesday, if you haven't attended an MCC event already, you can go to that and get your extra credit done, or you can just go by and get free stuff, because who doesn't like free stuff, right? So, yeah, I'll send out a reminder about that too. But they may have Starbucks on that day. I don't know. They'll be giving away free things. I know that. All right, so let's take a look at this figure again. As I was saying, these right here represent little bar graphs, okay? So the higher the bar, then the higher the partial pressure or concentration. So for instance, when we're looking at, for external respiration that we see here, we're looking at the air that we actually breathe in. So the air that we actually breathe in, when it gets to the alveoli of the lungs, the partial pressure, that's the P, the partial pressure of oxygen is about 104 millimeters of mercury. Now it starts out at about 160 like we just calculated. But remember that when we're breathing it in, it's going through this whole pathway that we just traced out here. And it's all lined with a mucous membrane, epithelial cells and stuff like that. And each of those cells are like, oh, I can use some of that oxygen. Hand me some of that, right? So they need to survive as well. So they're taking a bit all along the pathway. So by the time we get down to the alveoli, we go all the way through this, we get down to the alveoli, we see the partial pressure of oxygen has gone down a bit. It's still pretty high though, 104. We look at the partial pressure of carbon dioxide, that is pretty low, right? So it's about 40. Again, it's higher than in the air out here because all of those epithelial cells are also going, hey, take this. I don't want this carbon dioxide gas. Get this out of here, right? All right, so by the time we get into the alveoli, this is what we have for our partial pressure of oxygen and carbon dioxide. Now let's compare that to the pulmonary capillaries, right? So the blue, quote-unquote, blue blood that's arriving to the pulmonary capillaries from the right side of the heart, right? Right side of the heart sends it to the pulmonary circuit. Let's look at those partial pressures or concentrations. Partial pressure of oxygen is 40 millimeters of mercury, right? That's much lower than 104, right? There's definitely a good gradient there. When we look at partial pressure of carbon dioxide, it's at 45 millimeters of mercury, and you're like, that doesn't seem too high. But remember, here in the alveoli, it was 40. So in the pulmonary capillaries, 45 millimeters of mercury for carbon dioxide, and in the alveoli, 40. So there's a gradient. It's not as steep as oxygen, but there's still a gradient. So what does that mean? In terms of oxygen, we're going to see oxygen go from where it's a high concentration to a low concentration. So oxygen is going to diffuse from the alveoli into the pulmonary capillaries, right? So in terms of carbon dioxide, that's going to go the other way. Carbon dioxide is relatively high in the pulmonary capillaries, relatively low in the alveoli. So carbon dioxide is going to diffuse from high to low and go the opposite way, right? Now, in terms of that whole Henry's law and things like that, carbon dioxide is actually a little bit easier, a little bit more soluble, a little bit easier to go between a fluid and a gas. So that kind of makes up for the fact that it has a lower gradient like that. But what we see is that we see two gradients. We see oxygen is high in the alveoli and low in the pulmonary capillaries. So it moves into the blood, exactly the way we want it to go. Carbon dioxide is relatively high in that deoxygenated blood in the pulmonary capillaries, relatively low in the alveoli where we just inhaled this fresh air. So it goes the opposite way. It diffuses into the alveoli from the blood, exactly the way we want it to go. Okay? So that is external respiration. External respiration is exchange, gas exchange, between the pulmonary capillaries and the alveoli. And it's driven by diffusion, right? It's just diffusion and moving from one area where we have a higher concentration, though we call it partial pressure in this case, a higher partial pressure in one area and a lower in the other area. It goes from high to low, like a ball rolling downhill. Okay? So external respiration, we see that gas exchange occurring there. Now, while we're on this graph, let's look at our internal respiration down here. Internal respiration uses the same process, but it's between the systemic capillaries and the tissues instead. All right, so in the systemic capillaries, we have this well-oxygenated blood with low amounts of carbon dioxide in it arriving at the tissues. So let's look at our partial pressure gradient here. So in the tissues where we're actively producing carbon dioxide, we have a partial pressure of carbon dioxide of about 45 millimeters of mercury. We saw that before. And a partial pressure of oxygen about 40. Now comparatively, this highly oxygenated blood arriving there has a partial pressure for oxygen, 100 millimeters of mercury. And again, carbon dioxide is relatively low at 40 millimeters of mercury. So what do we get? The gradient for carbon dioxide is that it's relatively high here in the tissues because the tissues are putting it out, right? The ATP that's being made in the mitochondria, this is the byproduct from it. So these are active tissues that are creating ATP and they're cranking out carbon dioxide. It's like a little factory. So we have relatively high carbon dioxide partial pressure in the tissues, relatively low in the blood, and so carbon dioxide diffuses that way, right? Meanwhile, if we look at oxygen, it's going to go exactly the right way we want it to go too. Relatively high, like we saw here, relatively high concentration of oxygen in the blood, relatively low concentration of oxygen in the tissues, because the tissues are using that oxygen in order to do their aerobic respiration. So it tends to be low, right? So oxygen diffuses from an area of high to low and goes into the tissues. That is internal respiration. Like I said, you get a twofer, right? Respiration is gas exchange. It's based on a partial pressure gradient for oxygen and for carbon dioxide. They each have their own gradient, so they each go their own direction. And the question is, are we talking about between the alveoli and the pulmonary capillaries? That's external respiration. Or are we talking about the systemic capillaries and the tissues? That's internal respiration. That's it.
So if it's going from your extremities into your core, I guess, that's internal?
So now that sounds more like you're talking about gas transport, right? You're going from an extremity in the blood and you're taking it more towards the core of the body. That's gas transport. We haven't talked about that yet. We're really just talking about out here. We're talking about a muscle cell out here, and we're exchanging between a systemic capillary and that muscle cell, that gas exchange. That's internal respiration. Because there are two different internal sites, the blood, which is inside of us, and those cells that are also inside of us. That's
So and tell me again what external is?
External respiration is between the air in the lungs, in the alveoli, because technically that's still outside of us, and the pulmonary capillaries instead. So that's external respiration. Gas exchange with the external environment. Yep. So how does that seem?
Can you repeat the between the air and the...
So for external respiration, external respiration is gas exchange with the external environment between the alveoli and the pulmonary capillaries. So external respiration is between that blood in the pulmonary capillaries and the air in the alveoli. You can say air in the lungs as well, right? Because the alveoli make up the lungs. Yeah, that's okay. Now for these, you're looking at a lot of numbers here, and you're like, do I have to know 40? Do I have to know 105? Do I have to know 160 and all that stuff? I would say no. Okay? So I would say you do not have to memorize those exact numbers. What you do have to know is relative concentration, relative partial pressure. Where is it higher and where is it lower? Because that gradient tells us which way that gas is going to go, right? So if we're talking about down the tissues, the partial pressure of oxygen is higher in those capillaries and relatively low in the tissues. So it goes from high to low. It goes and diffuses into the tissues, okay? Meanwhile, carbon dioxide, it's the opposite. So you don't have to know the actual numbers, but you do have to know where it's relatively high and where it's relatively low. So you can say, partial pressure of whatever gas it is higher over here, it's lower over here, so it's going to go from high to low, and it's going to move this way. Okay? So for instance, if we were talking about, one last example, carbon dioxide, in this external respiration, we don't have to know actual numbers, but we would say carbon dioxide partial pressure is relatively high in those pulmonary capillaries and relatively low in the alveoli, so it's going to diffuse out of the capillaries and into the alveoli. You don't have to say the actual numbers. You don't have to say 45 millimeters of mercury, 40 millimeters. You don't have to know the numbers. Just be able to say where it's high and where it's low. That seem all right? Okay. So that means, so far, we have done this, right? We've done that, we've done that, we've done that. So we've done 3 out of the 4 different processes we have to know for this respiratory system. So now we finish up with gas transport, which we said should be relatively familiar because we talked about a bit of this when we were talking about the blood. Okay, so oxygen. What's the main way that oxygen is transported in the blood? It's mainly transported bound to hemoglobin, right? We talked about that. Red blood cells have hemoglobin inside. It's a 4-part molecule. Each one has a little binding pocket to it that contains iron, right? And so each hemoglobin molecule can bind 4 different oxygen molecules. So that means it has 4 pockets. So picture your pants or something like that. You got 2 pockets in the front, 2 pockets in the back. You could put oxygen in all 4 pockets, be walking around, be fully stocked with oxygen, right? So, in a way, you know, I always tend to think of, if you were fully stocked with something, right? I always think of our hemoglobin molecule as being almost like a philanthropist, somebody who has a lot of money or something like that, and they want to give some of that money away, right? So what they're going to do is they're going to go to their bank, they're going to fill their pockets with money that they're going to give away, right? Where it's a high concentration, if you will, of money. They're going to get it there. They're going to stock their pockets. Then they're going to go to the neighborhoods and they're going to give it away. And the poorer the neighborhood, the more they should give away, right? If they're going through a neighborhood and it's like, you know, the fancy neighborhood, they're like, you don't really need any right now. I think I'm going to hold on to it, keep going around and see if I can find a poorer neighborhood where they really need it, right? It's the same thing in terms of our hemoglobin as well. So up at the lungs, and we'll see this coming up at the lungs, that's like the bank. That's where we have the highest concentration of oxygen. We just inhaled it. And so that blood in those pulmonary capillaries, that's where hemoglobin's really going to restock. And it's going to bind more oxygen in those 4 little pockets of every hemoglobin, and it's going to restock. And then it's going to go out into the systemic circuit, and it's going to start unloading, giving it to the tissues that need it. So about 98% of our oxygen in the blood is bound to hemoglobin. About 2% of it or so is dissolved in the plasma. So most of it, yes, definitely most of it, is bound to hemoglobin. And when it binds to hemoglobin, it gives the blood that brighter red color. That's why venous blood, low oxygenated blood, is like a darker, kind of muddy red, if you will, as opposed to arterial blood, which is well oxygenated, is a bright red instead. The more oxygen we have associated with that iron, the brighter it gets. Okay, so we would say that hemoglobin is actually saturated when all 4 of those binding pockets are filled, right? It's full, totally full, saturated, can't take any more. Partially saturated when it's got an empty pocket or so, but it still has some oxygen. And so what is going to tell hemoglobin when is a good time to pick up oxygen and when is a good time to release it? Because again, like this rich person, what good is it just having this money if you never spend it? More so if you never give it away, right? So you should give it away at some point. And so it has to have some sort of indicator as to when is a good time to unload, when is a good time to reload. So these are some of the major factors that will influence whether hemoglobin loads up with oxygen or unloads oxygen. One of the main ones is partial pressure of oxygen. That makes sense, right? So I'm loading up oxygen in an area where there's lots of oxygen around. Again, like this philanthropist going to the bank, there's lots of money there. That's a good place to restock your pockets, right? There's lots of oxygen up in the alveoli in the lungs. That's a place, if there's a high partial pressure of oxygen there, I'm going to restock. I'm going to fill all those binding pockets, okay? But when we get down to the tissues, where it's a low partial pressure of oxygen, as it moves through those poor neighborhoods, it's like, oh, wow, here's an oxygen. Here's one of my 4. If it's really poor, here's my second one, something like that, right? So it's got 4, technically, that it can share. So partial pressure of oxygen is a major one that tells hemoglobin when to reload or unload that oxygen, okay, that it's carrying. Temperature is another one. And a lot of times when we look at these different factors that can influence hemoglobin, whether it's picking up or releasing oxygen, if you think about an active tissue, then a lot of those factors are going to have influence on these. A lot of what's going on in the tissue is going to have influence on these factors, which will then signal hemoglobin that it needs oxygen, right? So in an active tissue, we're going to have a low partial pressure of oxygen. We're going to have a relatively high temperature, right? Because think of like skeletal muscle. If you're exercising, it's getting warm. So that's a good signal. As the hemoglobin's coming by there, oh, you guys are working. That means you're probably burning ATP. That means you're burning through oxygen. That means I need to give you some, okay? So higher temperature, lower blood pH. We'll talk about things that influence pH, but you've heard of lactic acid buildup when you are exercising. So that can drop the pH in an active tissue, right? So if there's a drop in pH, then we're going to unload oxygen more. Partial pressure of carbon dioxide. Oh, okay. So in an active tissue that's making that ATP aerobically, it's going to be using oxygen and it's going to be releasing carbon dioxide like a factory. So if this hemoglobin swings into a neighborhood and there's lots of carbon dioxide being produced, oh, that means this tissue is very active. We're going to need to give you some oxygen because I know you're working hard. Okay? And there's a few other things. This one, BPG, is a metabolite. But the other ones that we see here, the first 4 that we listed, are critical ones in terms of influencing the binding of oxygen to hemoglobin or the releasing from it. Okay? And again, for these, you're not going to need to know an exact amount, but we're saying for each of these factors, if it goes up or if it goes down, how does hemoglobin respond? Does hemoglobin load up with oxygen or does it release oxygen to that particular tissue? I was going through it in terms of an active tissue, saying, oh, we're low oxygen and we have high temperature and we have low pH and we have high carbon dioxide. But if you have, say, a cold muscle that's not doing anything, then what's going to happen, right? It's going to have relatively high oxygen because it's not doing a lot, relatively low carbon dioxide, again, not doing a lot, low temperature, higher pH instead. And so all those indicate those cells aren't very active, so we don't probably need to deliver a lot of oxygen to it. So that hemoglobin might hold on to it instead. Okay? So that's what we actually see on these curves that we see here. These are called hemoglobin saturation curves. And what these are showing is at what times hemoglobin is going to release oxygen and which times it's going to load up on it. So if we look down here on the x-axis, this is showing the partial pressure, in this case, of oxygen, our most important factor for hemoglobin, the partial pressure of oxygen around the body from 0 to 100. And on the y-axis, we have percent oxygen saturation in hemoglobin. All right, so let's take a look at this. We'll start at the high point of the curve up here. So right here, we see that at 100 millimeters of mercury, that's one of the highest partial pressures of oxygen in the body. That's in the lungs. And so hemoglobin is 100% saturated. All 4 pockets are full. Up at the lungs, it says, hey, this is the bank we're going to restock. So I'm going to fill all my pockets with oxygen. So at our highest partial pressures of oxygen in the body, hemoglobin is going to be stimulated to reload and fill all of its pockets and become saturated. Now that hemoglobin goes out to the body, out to the tissues, and what happens, right? We see that the partial pressure of oxygen is going to drop because the tissues are using that oxygen. So let's say here, like we saw in that first graph, we get down to about 40 millimeters of mercury, common in the tissues. So if we go up here, what we find is that hits about this point on the graph. So we go over here, we find that that is about 75% saturation. Now 75%, if we change that into a fraction, that is 3/4. Hemoglobin has 4 binding pockets. So at the partial pressure of oxygen common to the tissues, at that stimulation of partial pressure of oxygen, our hemoglobin is going to release 1 out of its 4 oxygen molecules. Okay? So commonly, when the blood goes through there, the hemoglobins will go, oh, you look like you need some of this. This is kind of oxygen 4. Here's 1. Okay? Now, if you're really working hard and that partial pressure of oxygen in the tissues is even lower, then you might see hemoglobin go, wow, this is a really poor neighborhood. You're going to need 2, maybe 3 or something like that. But typically, as it goes in around the body, it might unload 1 out of its 4 different oxygen molecules. So you see it drop to like 75%. But this shows you the relationship between the partial pressure of oxygen in the tissues and the saturation of oxygen in hemoglobin, right? So again, if we go down, let's see, 50%, something like that, if we drop down to here in terms of partial pressure, we might give another hemoglobin away, or another oxygen away from hemoglobin. Okay? Now, it's a smooth curve like this because this is averaging like all the different billions of hemoglobin molecules, but we see that as we get into poorer neighborhoods for oxygen, hemoglobin tends to drop off more oxygen and release more oxygen. And as we get into richer neighborhoods, like the bank, like the lungs, it tends to then pick up oxygen instead and become fully saturated again. Okay? So this shows that relationship. It's not exactly linear, right? It's not a straight line between them, but it shows that relationship and the influence of the partial pressure of oxygen in the surrounding tissues and hemoglobin. Now there's pretty colors. Oh, that's nice, huh? So these ones are showing us the influence of body temperature. And we said that when there's a higher body temperature, because your body is working harder, hemoglobin is going to release more oxygen to those hard-working tissues. So what we see is that the temperature, 38 degrees is kind of average body temperature. When it gets hotter, we see this curve drop. So we see that curve drop. So like 75% on this curve here. If we look at it in a warmer environment, we can see that hemoglobin is now only saturated to about 60% instead of 75%. So what that means is, in a warmer environment, hemoglobin is like, oh my gosh, you're working hard. Here, have some more. Okay? In a colder environment, you see that curve goes up. That means it goes, you're not working too hard. You don't really need any of this. With these curves, with these saturation curves, you're not going to have to reproduce them or anything like that. You're not going to have to draw them or anything. But you still need to know that relationship. Like if this factor goes up or down, how does hemoglobin respond? Does it hold on to oxygen or load up, or does it release it? On the right, we see partial pressure of carbon dioxide, or pH. And so again, we see this drop in the saturation curve if the pH gets lower or if the partial pressure of carbon dioxide goes up, right? So those factors, if it shows that it's an active tissue, then hemoglobin is going to willingly give off that oxygen to that needy tissue. Okay. So that was about oxygen. Oxygen being transported mainly in the red blood cells, bound to hemoglobin. A little bit, like 2% maybe, that might be just floating around in the blood dissolved. And then we talked about some of the factors that will influence hemoglobin and its release or loading of oxygen. So I think there's probably an exam objective question that has to do with that. So that question is often written where it lists out the factors for you, like partial pressure of oxygen, temperature, and stuff like that, and you have to describe what happens in terms of hemoglobin. How does hemoglobin respond? Okay? All right, so now we switch over to carbon dioxide, Interestingly enough, carbon dioxide is almost the opposite. So most of the carbon dioxide in the blood is actually transported in the plasma. And if you remember when we were talking about blood way back when, we always say, don't forget about the plasma. There's a lot of important things that are transported in the plasma. Don't just think about the red blood cells and the white blood cells and the platelets. Plasma has a whole lot of important things to it, including most of our carbon dioxide. Now, interestingly, most of our carbon dioxide is converted into this, which is bicarbonate. And we'll look at the equation on how we do that coming up. But we will change carbon dioxide gas into that ion, bicarbonate, and that's the main way that it transports in the blood, floating in the plasma as a bicarbonate ion. Now there's also about 20% that is bound to hemoglobin. And you're like, how does that work? I thought hemoglobin just binds oxygen. Well, remember that it's got those 4 pockets. When it gets down to the tissues, if it empties a pocket of oxygen, now, hey, I got some room here. You got any garbage for me to pick up? Here's that. Now I got some room. I'll take some garbage. I'll put that in my pocket. So hemoglobin can actually transport carbon dioxide as well when it has a free pocket. Okay? So about 20% of our carbon dioxide is transported attached to hemoglobin. Carbaminohemoglobin is a term for when it has carbon dioxide attached to it. And then about 10% is actually just floating around in the plasma itself. So the primary way that carbon dioxide is transported in the blood is in the plasma as bicarbonate ion, and we'll look into that coming up more.
Oh, so for when it takes away the carbon dioxide, is it the iron that's going to be attaching for it to stick on as well?
Yep, so it's still in that little binding pocket where oxygen would normally sit. It's still in the pocket. Yep. Now here's another interesting thing related to that binding pocket. Carbon monoxide, right, from combustion and stuff like that, that can actually asphyxiate us and we can die. That also can bind to hemoglobin. And here's the wacky thing. Carbon monoxide, hemoglobin itself will bind to carbon monoxide more than carbon dioxide and more than oxygen. So if it has a decision, it's like, ooh, who wants to be carried? It's going to choose carbon monoxide over oxygen every time. That is bad, right? So that's the problem. Like carbon monoxide, you know, there are other products in terms of combustion and fumes and stuff like that, but carbon monoxide, you may not be aware when carbon monoxide levels are high. Right? That's why you are recommended to have a carbon monoxide monitor in your house. You usually mount it somewhere near your garage because that's where a lot of them come from. So you should have one in there because when carbon monoxide levels get high at all, it starts taking the place of oxygen in your hemoglobin. Now what happens is your red blood cells get to the tissues, and it's like, here, you want some of this carbon monoxide? It starts starving your tissues of oxygen because it's had carbon monoxide in all those different pockets instead. So it can cause you, right, to pass out or something like that, or even die. So when you see in dramatic movies or something like that, somebody's trying to commit suicide, and they take a hose and they go from the exhaust of the car and they pipe it into the car, that's because carbon monoxide starts to take the place of all the oxygen. Even though they're breathing, it starts to take the place of the oxygen in that air and starts starving the body of oxygen. So, don't do that. The percentages, I would know. I mean, you could, if you wanted to change, 2 ways, right? So you could do numbers, if that's easier, or you could do mostly in the plasma as bicarbonate, some attached to hemoglobin, and a little diffused in the plasma. You could do that. But this is the critical one, that this is the main way that carbon dioxide is transported in the blood. Yeah. So it's up to you, whether the numbers or just kind of that relative description is easier. So this is the equation that I was talking about. You do need to know this equation, okay? I know it's not chemistry class or whatever, but this equation will come up again and again. We'll see it in the urinary system, we'll see it in the digestive system, etc. Lots of cells in the body use this equation in order to, in this case, change carbon dioxide into bicarbonate ion. So if we take a look at this, what happens is, as our carbon dioxide comes from the tissues and it goes into the blood, it combines with water. So we see CO2 plus H2O transiently, temporarily gives us that, carbonic acid, right? So you can see there's 2 H's and 3 O's and 1 carbon, right? 1 carbon. So everything adds up. So temporarily it gives us carbonic acid. We know that an acid, when you take an acid and you have it in an aqueous environment, what happens? It falls apart into something plus a free hydrogen ion. That's the definition of an acid, right? It gives off electrolytes, but one of them is a hydrogen ion. So carbonic acid quickly falls apart into a hydrogen ion, and then, ta-da, there's our bicarbonate ion. Okay? So as the carbon dioxide comes from the body tissues into the blood, we're doing a quick change and conversion into bicarbonate using this equation. And there's an enzyme that facilitates that. Now here's the funny thing. So here's the name, carbonic anhydrase. Notice, right, ASE. It's got that suffix that helps us to know that it's an enzyme. Okay? Carbonic anhydrase facilitates this equation. Now look, it does go both ways. Like we see like that. It's a reversible reaction. But here's the wacky thing. That occurs in the red blood cells. What? So bicarbonate ion is mainly transported in the plasma, but this chemical conversion is occurring in the red blood cells because it's inside of the red blood cells where we have that protein, which is an enzyme called carbonic anhydrase. So we do a quick changeover from carbon dioxide into bicarbonate ion inside of the red blood cells, and then we're going to release that into our fluid, into our plasma. So this is what we see right here. So here, tissue cells, where we're cranking out all this carbon dioxide, right, like little factories. It diffuses into the interstitial fluid. It diffuses into our capillaries. So these are simple squamous epithelial cells that you see right there, right? So we see this big diffusion here, and it goes into the red blood cells. So it goes into the plasma, but it quickly goes into the red blood cells. There's our carbonic anhydrase that combines the carbon dioxide with water, makes our carbonic acid temporarily, and now we see it falling apart into bicarbonate ion and a hydrogen ion. So the bicarbonate ion is then transported out into the plasma, right? Because we said bicarbonate ion is transported in the plasma, the main way that it's going around in the blood. And that hydrogen ion oftentimes is bound with hemoglobin. So that's like a fourth thing that hemoglobin can bind. It can bind oxygen, it can bind carbon dioxide, it can bind carbon monoxide, and it can bind hydrogen ions like that to help stabilize pH. Because if the hydrogen ion isn't floating around on its own, then it's not making things acidic. So if we can bind it up with something, in this case hemoglobin, it keeps things from getting too acidic. Okay? So that, I think, is always kind of a wacky thing to me, that we talk about carbon dioxide mainly being transported in the plasma, but the conversion that makes that possible, that changes it into bicarbonate ion, actually happens inside of the red blood cells. So now we have this wacky thing. We're like, okay, hemoglobin transports oxygen. I got that. I got it. Oh, but it can transport carbon dioxide as well, right, when it has a free pocket. And all of a sudden, red blood cells have a role in terms of converting carbon dioxide as a gas into bicarbonate ion so they can float around in the plasma as well, right? So it's got this interesting role in many ways. And then on the other side of this, right, we see oxygen being released from our hemoglobin, and then the oxygen diffusing into our cells like that. And when it separates from hemoglobin, that's why now hemoglobin has a free pocket to bind up that hydrogen ion. Crazy, right? It's like all of a sudden it's like very efficient. It's like, hey, I just dump this off, I can pick this up, and I can do it over here, and I can exchange it over there. And all of this while we're flowing through those capillaries. That's why we need to be nice and slow, right? We talked about that in the cardiovascular system. If it was whipping through there, it wouldn't have time to do all this conversion and change and stuff like that before it ended up in a venule and now it's gone, right? So this is what happens down at the tissue level. This is what happens up at the oxygen. This is external respiration. So this is external respiration right here because it's between the blood in the pulmonary capillaries and the alveolus. So this is an alveolus, and this is the blood in a pulmonary capillary. So in this way, what we do is we take that bicarbonate ion that is floating around in the plasma, and now we're going to use that same equation, but we're going to go the opposite direction instead. So we see the bicarbonate ion, we pull it into the red blood cell, we combine it with hydrogen to get carbonic acid, and then that carbonic acid, using the enzyme, we change it into carbon dioxide and water. And then the carbon dioxide is going to diffuse out, diffuse into the alveolus, partial pressure gradient, and now we can exhale that carbon dioxide, right? We can't exhale bicarbonate ions, but we can exhale carbon dioxide. So we got to switch it around, and we have to convert it back the other way when we get up to the lungs. And that carbonic anhydrase can work both ways. So it's a reversible equation that you have to know, that water plus carbon dioxide gives us our carbonic acid, which then falls apart into a hydrogen ion and a bicarbonate ion, and or the reverse of that, right? It can go the other way as well. So these are all factors that help us to understand how those gases are transported in the blood, right? In terms of hemoglobin, in terms of this carbonic anhydrase and converting most of our carbon dioxide into bicarbonate ions. Again, we do see some that's dissolved in the plasma, things like that. But that is the main way that this works. Finishing up, there are some other factors that come into play in terms of these processes that we see here. So the amount of, and much like we saw before, the partial pressure of carbon dioxide affects the loading and unloading of oxygen. And in fact, there is the opposite effect as well, the Haldane effect. I wouldn't really worry about that. I wouldn't worry about that. Don't worry too much about that. We'll talk about that a little bit later on. But we just know that there are factors that can affect these processes because the demand of the body needs to be communicated, if you will, to things like hemoglobin, to things like these enzymes that are going to work to provide what the body needs. Okay? Okay. So that carbon dioxide, like we said, you don't need to know it's called the Haldane effect or anything like that, but the carbon dioxide that we have, because of this equation, actually affects the pH, right? Because every time we convert carbon dioxide into carbonic acid into a bicarbonate ion, we release a hydrogen ion. Now, hemoglobin can bind some of those up, but some of that hydrogen ion is going to just get loose as well. So the more carbon dioxide that we have, because the tissue is really active, the more acidic that tissue becomes. So not only from lactic acid like we may have talked about before, but that carbon dioxide itself, through this conversion, makes the tissue acidic too. So every time the partial pressure of carbon dioxide goes up, the pH drops. It gets more acidic associated with that. So that's why, in fact, a lot of times when we're going around the body, we actually monitor, we have, in essence, pH receptors around the body that are monitoring the pH because we use that as almost a surrogate for carbon dioxide levels. Like if the pH is dropping, that means the tissues are really active, and so, right, we may need to change breathing rate, depth of breathing, or the frequency of breathing to get more oxygen in there, to get rid of that excess carbon dioxide, etc. So these act as stimulators for our regulatory processes, right? So when we look at breathing, if we need to change breathing, it's the areas of the brainstem that control breathing. Our medulla and our medulla oblongata and our pons are the main areas of the brainstem that control breathing. Now, to a certain extent, the medulla oblongata, the lower area, is in direct communication with the lungs. So this area here, the medulla oblongata, is the main area of the brainstem that controls breathing, your regular breathing, like when we're doing breathing in, breathing out, tidal volume like that. The pons itself kind of oversees it and does kind of specialized functions, like if we're doing like forced inhalation or forced exhalation, or when we're switching over from inhalation to exhalation. Those types of more complicated things, we need like higher management to overlook those or kind of oversee those, and so the pons comes into play there as well. But for the most part, those are 2 areas of the brainstem that control breathing, both depth of breathing, breathing rate, and things like that. Now there are, if you look in the notes, there are these specific different areas in the pons and stuff like that, the dorsal respiratory group and the ventral respiratory group and stuff like that. Don't worry about that. Okay? So if you see any of those, and it says the pre-Botzinger and stuff like that, any of those specialized groups of neurons and things like that, don't worry about that level of detail. Just know pons and the medulla oblongata are the main areas of the brainstem that control our breathing. So they're getting information from our chemoreceptors, peripheral chemoreceptors out in the body, which tells us, oh, hey, look, for instance, there's a drop in oxygen, there's an increase in carbon dioxide, there's an increase in hydrogen ions, right? From the peripheral or the central chemoreceptors associated with the brain, if we see increases in the activity of our muscles and joints, if there is something that is emotional or something like that, or if we're having pain, right? If you hit your thumb with a hammer, you go, like that, right? That has an influence on breathing all of a sudden, right? So all these things can increase our breathing rate. If you look on the right side, we see things that will decrease breathing rate as well, right? So any of these factors come into play in terms of monitoring the respiratory system and changing what the lungs are doing at any particular time based on what the needs of the body are. Okay? So our breathing rate should match what the body's doing. Now, if it goes too far, then we can get into situations where things get wacky, right? So like hyperventilation, too much ventilation, is when your breathing depth and rate is more than you need for what the body is doing. Like when you're running, I did my last run of the season this past Saturday. We did the Nun Run. That's a cool run. So it benefits this local cloister of nuns. And they do a 5K and a 10K. And the 5K, some of the nuns actually run in their wool habits. That's interesting to see, right? They're like full on in their whole garb running the 5K. So you feel bad when you beat them, but not as bad you're going to slow down or anything. Anyway, I was doing the 10K. Point being, when I was running, my breathing rate was really high. Like my heart rate was like 170 or something like that, uncomfortable. And my breathing rate was really high because it was matching my bodily demands, right? I was cranking out a bunch of carbon dioxide. I was using a bunch of oxygen, those muscle cells. So that's an increase in ventilation. It's not hyperventilation. Hyperventilation is if you think about the up-and-coming test, and you start having a panic attack, and you start breathing more, but you're just sitting there, right? That's hyperventilation. So you don't need to do that. Hyperventilation is going to try to get rid of extra carbon dioxide, more so, and it may lead, it comes from hypercapnia. So hypercapnia is where we have too much carbon dioxide, right? And we may need to get rid of it. So for these terms, you should be able to just define these terms. You should find them in the vocabulary list. Don't forget to review your vocabulary list because there will be vocabulary questions. Hyperventilation, hypercapnia, hypoventilation, slower breathing rate. Apnea has to do with basically your breathing stopping temporarily. You hear about sleep apnea, right, where people stop breathing at night for whatever reason. Their pons and medulla is not monitoring and adjusting the situation accordingly. So it's letting it drop too low and even stop it at times. So hyper and hypoventilation, you should know capnia, right? Remember that's CO2, so there's hyper and hypocapnia. There's also, just to make sure you know that, hypoxia. Remember, hypoxia is low oxygen. So if we get low oxygen levels or something like that as well, we may increase breathing rate in order to bring those oxygen levels up. Okay? Okay, so that takes us to the end of respiratory system, just about on time. So what that means is that on Wednesday, we're just going to be doing review. That's the whole reason why we do exams online, is so that we can have, rather than doing an exam on Wednesday, we can have time together to go through. So make sure that you're looking over everything. Look over your exam objectives, all of your notes and things like that. Come with questions. So start studying, start reviewing things, start talking to each other, and come with questions so that when we are going through things, we can be answering the questions that you have, so that we can make sure you have everything you need so that when you're studying for that Saturday exam, you're all ready. Okay? All right. So that's going to be where we stop for now.